talk about the cell cycle. So what is a cell cycle? Cell cycle is the stages through which a cell passes from one cell division to the next. And to get through this, we will first define what is a cell. Cells are the basic building blocks of all living things. The human body is composed of trillions of cells. They provide structure for the body, take in nutrients from food, convert those nutrients into energy, and carry out specialized functions. Cells have many parts, and each with a different function. There are two types of cell cycle. First is the mitosis. Mitosis is the process of making new body cells. And then the meiosis. The meiosis is the type of cell division that creates egg and sperm cells. In mitosis, there are two phases. First is the interphase. Interphase prepares the cell for division, and it is composed of three different stages. First is the gap 1, second is the synthesis, and then the gap 2 phase. Second is the mitotic phase. This is where the cell division process actually occurs. Process of cell duplication in which one cell divides into two genetically identical daughter cells. Mitotic phase composed of four different phases: prophase, the metaphase, the anaphase, and the telophase. We also inserted here the cytokinesis. Cytokinesis is not actually a part of the cell cycle, but considered the final stage of cell division, which the cytoplasm splits into two and two daughter cells form. And now we will proceed to the meiosis. Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. In meiosis, it is divided into two parts, the meiosis 1 and the meiosis 2. In meiosis 1, chromosomes in a diploid cell resegregate, producing four haploid daughter cells. It is the step in meiosis that generates genetic diversity. Just like in mitosis, meiosis also have four different phases. These are the prophase 1, the metaphase 1, anaphase 1, and telophase 1. Once meiosis starts, the purpose is to produce a haploid gamete, so there is no further need of replication growth. Hence, between meiosis 1 and meiosis 2, there is no interphase. After the meiosis 1 is the meiosis 2. In meiosis 2, the sister chromatids within two daughter cells separate, forming four new haploid gametes. The mechanics of meiosis 2 is similar to mitosis, except that each dividing cells has only one set of homologous chromosomes. In meiosis 2, there are also four phases: the prophase 2, metaphase 2, anaphase 2, and telophase 2. The first round of nuclear division that occurs during the formation of gamete is called meiosis 1. It is also known as the reduction division because it results in cells that have half the number of chromosomes as the parent cell. While meiosis 2 is known as the equational division, as the cells begin as haploid cells and end as a haploid. Thank you for listening to this brief discussion about the cell cycle. And to enlighten you more, I will give now to the next reporter to discuss to you the meiosis and mitosis and what really happens during these phases, during this cycle. Thank you.
Mitosis and cell division.
Now, I'm going to discuss cell division. So what is cell division? Cell division is a division of a cell into a daughter cells with the same genetic material. Means it is the process of reproducing another cell. Cell division has different phases. It calls cell cycles. Cell cycle is the series of events that cells go through leading to cell division. It has many processes undergo before it will eventually divide and reproduce a copy of a cell. The most important stage here in the cell cycle is the S phase or the DNA replication, because in DNA replication occur the photocopying of chromosomes when duplicating the cell. It reproduces equal amount of chromosomes. Other stages or spaces is G1, G2, and S, which collectively calls interphase. As you can see in the interphase, there's a presence of chromosomes. Also, there's a thread-like distance chromatin enclosed by nuclear membrane. Now, when the cell is ready to divide, we'll move on to the next phases, which calls mitosis.
What is mitosis? Mitosis comes from the Greek word mitos, meaning thread. In 1882, the German biologist Walter Fleming described a thread-like chromosomes that mysteriously appeared in animal cells just before they divided in two. Mitosis is a process of nuclear division in which the replicated DNA molecules of each chromosomes are faithfully partitioned into two nuclei. Mitosis is usually accompanied by cytokinesis, a process by which a dividing cell splits in two, partitioning the cytoplasm into two nuclear packages. Mitosis is generally divided into 5 stages: the prophase, prometaphase, metaphase, anaphase, and telophase. Prophase. The first stage of mitosis. The duplicated chromosomes are prepared for segregation and the mitotic machinery is assembled. Events in prophase. During prophase, the chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. The cytoskeleton is deassembled and the mitotic spindle is assembled. Prometaphase. The dissolution of the nuclear envelope marks the start of the second phase of mitosis, or the prometaphase. At the beginning of prometaphase, compacted chromosomes are scattered throughout the space that was the nuclear region. Events during prometaphase. Chromosomal microtubules attach to kinetochores of chromosomes. What is a kinetochore? Kinetochore assembles on the centromere during prophase. The kinetochore function as a site of attachment of the chromosome to the dynamic microtubules of the mitotic spindle. Also, the residence of several motor proteins involved in chromosome motility and a key component in the signaling pathway of an important mitotic checkpoint. After the attachment of chromosomal microtubules, the chromosomes are moved to a spindle equator. Once all of the chromosomes have become aligned at the spindle equator, with one chromatid of each chromosome connected by its kinetochore to microtubules from one pole, and its sister chromatid connected by kinetochore to microtubule from the opposite pole, the cell has reached the stage of metaphase. Events during metaphase. The plane of alignment of chromosome is referred to as the metaphase plane. The mitotic spindle of the metaphase cell contains a highly organized array of microtubule that is ideally suited for the task of separating the duplicated chromatid positioned at the center of the cell. The fourth stage is the anaphase. Anaphase begins when sister chromatids of each chromosome split apart and start their movement toward the opposite poles. As the chromosomes moves during anaphase, its centromere is seen at its leading edge, with the arms of chromosomes trailing behind. Events during anaphase. The centromere splits and the chromatids separate. Chromosomes move to opposite spindle poles. The spindle poles move farther apart, and the movement of the chromosomes toward the poles is referred to as anaphase A to distinguish it from a separate but simultaneous movement called anaphase B, in which the two spindle poles move farther apart. As the chromosomes near their respective poles, they tend to collect in a mass, which marks the beginning of the final stage of mitosis, which is the telophase. Events during telophase. Chromosomes cluster at opposite spindle poles. Chromosomes become dispersed. The nuclear envelope assembles around chromosomes clusters. The Golgi complex and endoplasmic reticulum reforms. And lastly, daughter cells formed by cytokinesis.
Let us all listen and sing along with the mitosis song.
Well, if the process of mitosis leaves you in a daze, then you need to focus on each separate phase. The letters P, P, M, A, T will help you memorize them individually. Prophase, prometaphase, metaphase, anaphase, and telophase. In prophase, the chromosomes suddenly appear by condensing into chromatids with centromeres. Pairs of chromatids, like tiny coiled ropes, are visible if you look through a microscope. Prophase, prometaphase, metaphase, anaphase, and telophase. In prometaphase, that's the nuclear membrane, breaks down microtubules from the centrosomes invade, probing at the center of the chromosomes for the centromeres where they attach to kinetic cores. In metaphase, the chromosomes line up on a plate at the cell's equator, where they wait for the next phase called anaphase to start, which is when the paired chromosomes are pulled apart. Prophase, prometaphase, metaphase, anaphase, and telophase. In telophase, the separated chromosomes arrive at the poles where they are surrounded by reconstituted nuclear membranes, uncoiling and diffusing into chromatin again. Prophase, prometaphase, metaphase, anaphase, and telophase. Prophase, prometaphase, metaphase, anaphase, and telophase.